Hi, this is Mike Lawless, and in this video, I'll discuss the population simulator in GastroPlus. The GastroPlus population simulator enables you to simulate a population of up to 2,500 subjects. It produces randomly generated subjects by varying selected or all physiological model, compound, and formula parameters using a Monte Carlo approach. This enables you to modify gastric transit times, pH, fluid volumes, pharmacokinetic parameters, PBPK parameters, dosage form, and compound parameters. The populations can be saved and reused for crossover studies. GastroPlus 9.6 has significant improvements in the population simulator, including covariant tissue models, but these only account for intersubject variability. This diagram shows how you can define different age, body weight, and BMI ranges for the population. On the left, I show ages ranging from 25 to 45 years old, and on the right, from 6 months all the way to 2 years old for more pediatric type simulations. This slide illustrates the generation of virtual populations using the previous algorithm that would be in GastroPlus 9.5 in previous versions. Here, we randomly select an age and gender from a defined population and create a typical physiology for that subject. Next, random variability is added to the individual tissue sizes, and this is centered around the typical tissue size for a given age and gender. Then we update the body weight for the virtual subject. This next slide shows population generation for 500 subjects from 2 to 40 years old with 50% males. And you can see that in the body weight and the body height that there's a pretty narrow distribution of values. For example, for the ages up here, it's fairly tight until you get to around 18 years old. Then it starts separating a little bit, but still it's a fairly tight band, similarly with the body height. There's no variability in the body heights for the given population. This slide shows the new algorithm. So here we select an age and gender from a defined population. Then we select the body weight and height based on a bivariant distribution for a given age and gender. So this is this little plot here. Then we generate the physiology with the tissue sizes corresponding to selected age, gender, height, and weight. And there's also an option to use additional random variability on the tissue sizes as in the previous algorithm. This slide shows the same number of subjects and ages as the previous slide with the previous version of the software. And here you see that the body weight variation is much larger. And also there's not these gaps in the body height. So we generated a much better variability on this, and there's more options for creating populations of different subjects. This next slide shows just that the more subjects you have, the wider the variability. So the red points here are just for a population of 50 subjects, whereas the other one was 10 times as much. Once the simulations are performed, the results of the trial are presented in two tables. On the top table, the mean of the various PK parameters are shown, and then on the bottom, these are the geometric mean values for these. Experimental plasma concentration time profiles found for the record. This can either be an IPD or an OPD file. Then the results will be colored according to the points over here on the right. In the upper table is the arithmetic mean. The green font means that the simulated 90% confidence interval is within 80 to 125% of the bioequivalent limits. Red font means that they're not in that stage. And then similarly for the lower table, you have a similar coloring scheme, except it's looking at the geometric mean of the Cmax and AUCs. You can also create plots, and the new plot this is an example of a virtual trial of about 200 subjects. The blue line here is the mean of the 200 subjects, and then this green band is the 90% confidence interval for those subjects. And then these light blue ones get further and further probability limits there. Now I'll demonstrate the population simulator interface in GastroPlus 9.6. I have a single record in this database, and it's also associated with an OPD file with some data that I simply made up just for this demonstration. If I go to the Simulation tab, you'll see that there's a button called Population Simulator. If I click on that, that will highlight it and bring up the Population Simulator dialog box. All parameters, except those with a baseline value of 0, are selected by default. These are all the parameters that can vary in a population. Coefficients of variation and distribution types are also shown in these last two columns. One can click on the Set Pair button to define your population. So you can vary the sample size. You can go from healthy or various disease states. You can adjust the percent of males, the age, and the ages will go from months, weeks, days, as little as a few days, and years, and other weights and BMIs can also be adjusted with this dialog box. Once you have the populations defined, you can save them under the File Save button. After setting up your population, you'll be returned to the Simulation tab in GastroPlus. Note that the panel containing the color image of the GI tract has been replaced with a warning label suggesting that it would be wise to decrease the number of output points. Otherwise, the population simulations will run unnecessarily long. To correct this, go to the Simulation Setup and click on the Output Data Points and reduce that to a more reasonable number like 25. Once that is fixed, you'll return to an empty population simulator results table. On the left-hand side here, you can set the sample size. I'm going to set this to 100 samples, and then click the Start button to start the simulation. Here, you'll be prompted for the stochastic run save file, and it has a default as the compound name and then the number of subjects you're running. And then I'll just click Save. Next, you'll get a warning message here that the KP depends upon some of the parameters that were selected, such as log P, FUP, etc. But in some of the tissues, it was also selected for independent analysis and optimization. And this dialog box just asks you if you want to update the KPs in the tissues, which are not selected independently. I'll go ahead and click Yes. And then another dialog box will come up saying that you've selected absorption scale factors in the population simulator. So now it will go back to the user-defined ASF model. Once I dismiss that dialog box, the simulations will start, and you'll see this number incrementing until it gets to 100, at which point it will finish. So we'll just let that go ahead and finish running. Once the simulation is finished, the table will be populated. So you get the fraction absorbed, fraction delivered to the portal vein, oral bioavailability Cmax, Tmax, area under the curve going to infinity, and area under the curve going to the specified time, which was 24 hours in this case. The mean value will be displayed along with the geometric mean, so you get the arithmetic and the geometric mean, the correlation of variance, the minimum and maximum values for each one of the parameters, and then the 90% confidence intervals for the untransformed data, and then the natural log transformed data. There's also additional information in the .stc file. Next, you can go into the Graph tab and click on New Plot. This will bring up a plot of the results. The blue line here, as mentioned in the slides, is the mean for the 100 subjects. The green band is the 90% confidence interval, and then these blue or cyan dots are the observed mean. Which again I entered kind of randomly just for this demonstration. On the left-hand side, there's a button that says Show Probability Curve. So when we click that, it will show all the probabilities for the 100 to 25% confidence interval. To clean this up a little bit, we can display the legend on the right-hand side and then resize the window in order to make the graph look a little bit bigger. This concludes this video demonstration of the population simulator in GastroPlus. If you have any questions, please email me at mlawless@simulations-plus.com. Thank you.